It's a real pleasure to be here. I arrived late last night, but I'm going to stay until the end. So I'm going to be available. If you want to interact with me, I will be around until the end of the school. Okay, so I have a double position, and I have a double kind of. So I do work as a scientist, and you will see it, protocell modeling and things like that, in vitro, in silico. But I also keep a track that is more conceptual, more philosophical. And I'm going to show you in this, probably in the second part of the talk, about how the two things can go together, actually. But in the first one, in the first part, I will give an overview of the field of origins of life, so that you have an idea and how things are changing quite a lot and quite in promising ways. Okay, and it's in particular this systems approach that some chemists have taken up that makes a big difference in the 21st century, and I'm going to try to tell you what it is about. Okay, so on the one hand, I mean, the good thing about combining philosophy and science is that, okay, we have to be somehow so proud of the knowledge that humans have about the world, okay, because it's incredible that we understand so much. But at the same time, we shouldn't be too arrogant, okay, because when you see the complexity of the world out there, you realize that you don't understand a lot more than what you understand. Okay, so philosophy is always good to tell, you know, these scientists that are a bit too arrogant that there are questions, big questions that haven't been answered, and so in this way, it has a critical view on scientific advances that is very healthy to keep. Okay, so you. But I will make the point that philosophy has even more things to contribute to science if philosophers are involved in the actual designing of experiments and interpretation of experiments, and they get involved into, you know, it's not philosophy of science, we call it philosophy for science or philosophy in science. Okay, so it's more involved in the actual development of scientific models and theories, and so. Philosophy, maybe we hear a bit more tomorrow, but it has a role, also a constructive role, and particularly if you're dealing with very complex systems like biological systems, where you need to have different approaches to actually get an overall idea of how they work. These different approaches, these levels of description that you have in the system, need a language to actually be interrelated. Okay, and that language comes from natural language, but it's a kind of a specific type of language that you have to develop. And philosophers, you know, if they do a good job, are able to generate that kind of language with metaphors. But all science is full of metaphors anyway. So, but we have to take this very seriously, because. As some philosopher, and this is not a philosopher, he's a physicist in fact, but he was claiming that this kind of natural language, this is Walter Elsasser. This is I would like to start my lecture with a quote from this physicist that says like that: if relationships become too complex, conceptual expression may well be the most appropriate way of formulating certain aspects of the theory, and given the inhomogeneity of many classes of biology, conceptual language, okay, may frequently be the most suitable and appropriate means of expressing the basic relationships pertaining to the regularities, the principles, the laws of biological theory. So here is a physicist that doesn't have a reductionist approach, but he says, okay, we need to formalize, we need to quantify. That's the main effort of science, but at the same time, conceptual language is going to be helping us to understand as well, so we shouldn't put it aside. Another quote from a physicist, more famous than Elsasser, is Richard Feynman. And Richard Feynman was taking the opposite. So sometimes when you look at things from a philosophical or from a more poetic or more conceptual or more whatever, there is some criticism of science for being reductionist. And Feynman was given a reply to this, saying, well, it's a long quote. I will not read it, but it's just someone watching a flower and how beautiful it is. And if you try to put it into pieces, then you destroy the beauty of the flower. Blah 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 blah. And Feynman says, no, I'm a scientist and I can enjoy the beauty of the flower as well, but there are many other things that we can add and many other aspects of it if we put science there. Okay, so here comes the short passage. There are all kinds of interesting questions that come from a knowledge of science, which only adds to the excitement and mystery and awe of a flower, right? It only adds, he says. I don't understand how it subtracts. So but you have to, so the thing here is science is the most elaborate and reliable type of knowledge that humans have created, but then we have to kind of put it in context, see the limitations and all the rest of it. Okay, and natural language, we should not, natural language, all scientists use, all scientists, even all experimental papers are simply weak papers because they just report experiments. When you combine them with theory, when you combine it with, then they become stronger. So and in each paper that you read, there will be always language, natural language being used, okay, with ideas around the experiments, ideas about the theories. So we should have a kind of an open mind, okay, and this is kind of the general setting from which I start. Okay, so we're going to be dealing with origins of life, and the from an astrobiological point of view, which is the one that I should take here, I think it's very interesting that because it poses the problem in general terms, but unlike other approaches like artificial life or other kind of more abstract approaches, I think that it has the virtue that it does it in realistic physical-chemical terms. Different planets, different conditions, maybe different solvent, maybe different, organic compounds. But there is, and here I'm starting to be provocative, the field, I think, is still under the shade of reductionist approach. It comes from molecular biology, but also from chemistry, okay? And all these guys that get, maybe I'll be provocative from the very beginning. They get super excited when they see an organic molecule out there in the space, and a new molecule, and you have a spectrum. Okay, it's perfect, very nice, but it doesn't have anything to do with life, okay? Life is not organic molecules. It's a game that is played by organic molecules with inorganic molecules. Water is inorganic, okay? We are 70% of us living beings is so getting crazy about the molecules that you find in space or is fine, but has nothing to do with life, okay? Life is someone is laughing there. Maybe not everybody is convinced, but I will try to show you that. That's the case, okay? And we had a great talk before mine. That it was an introduction to different types of microbes. Last night when we had this, we were watching this documentary. I was already making the point there. Basically, if you see in this timeline. So there's the time window for the origin of life, origin of unicellular organism, is some tens or 100 million years, when the Earth, cooled down, you had water available, and then basically the first traces of microfossils tell you that around 3,500 million years ago, okay. We could more or less, there's a lot of debate about it, but I will not enter that debate. Okay, more or less, or roughly, pretty soon after the Earth cooled down, pretty soon in geological time, which is not chemical time or biochemical time, but pretty soon in those huge scales of geology, life appeared. Okay, but if you see here, most of the time that life has been on the planet, as I was saying yesterday, has been in the microbes. Okay, it took a long time of evolution. Okay, to the first traces. Again, there is a lot of debate here on where we have let's say the emergence of eukaryote cells, which are complex cells, as Anais was explaining before. But it took, you see, a long time, much longer than the actual time window for the origins of life. Okay, so, and then plants and animals and things like you were seeing in the documentary last night are really late comers. Okay, and here there was already a biosphere. Okay, and everything was set up. Okay, and so you have to think about the basic phenomenon as a phenomenon. Life is a microbial phenomenon. Okay, then microbes did excursions into more complexity, and in particular the emergence of eukaryote cells is super important to understand how complex forms of organization like us, multicellular organisms with cognitive capacities and stuff like that, can be around. But they are like, you know, how was the term for that? Like, you know, well, okay, it's like expressions of life at the basic level now that have been like curlicules. Do you understand the term curlicule? Like something pretty and nice and that, but the basic phenomenon is at the microbial level. Okay, so these are this should be the actors in the film that we saw yesterday, as I was saying. Okay, and if you go somewhere else in some other planet, I'm pretty sure that there will be cellular organisms like this. Life has, you know, this set. I think that all life that we will ever find anywhere should, for reasons that maybe I can go into a bit later, should be cellular. Okay, and there's going to be diversity in the population. There are going to be interactions that are ecological, diverse types of interactions at the ecological level. And these populations will evolve in time through, you know, time scales and space scales that are well beyond the time scales of each of these single organisms like we are. Now, we are individuals, we are here for a while, and then life persists. Okay, so the phenomenon of life is expressed in individuals, in organisms, okay, here unicellular organisms, but it clearly goes beyond that. Okay, it has an ecological and evolutionary dimension, you know, that goes beyond individuals. Okay, so that's something that I will come back to at the end of my talk. But if we see what's within, I'm going to show you the level of complexity of each of these individuals now. Okay, and how that cannot come but from a process of evolution. Okay. So if you take one of these microbes and you see inside, the first thing that people typically don't highlight, but is very, very remarkable, is that inside this, the first close-up, inside these microbes, water is not in the same state in different. So you have some areas. Maybe I can use the pointer here. There are some areas here that you see can be taken as a water solution. Okay, here are the ribosomes, the ones responsible for protein synthesis, and then some other areas that are more gel-like. So there's a lot of heterogeneity within each cell. Okay, and water behaves differently at different areas of the inner part of these cells. Okay, this is the first striking thing that people typically don't highlight, but it's very important. And then you have the membrane, and then, okay, if you do a bit of another close-up a bit farther down, farther deeper, you can see that you have the membrane here. You have this area here where you have these aqua solutions where you carry out metabolism, and then you have all this part here, which is this part here, which is full of macromolecules like DNA and proteins, and you know, there's a lot of macromolecular crowding here that makes these processes behave completely differently as the ones that we see here. okay? But all this machinery and all this membrane and all that is generated, is produced by the system itself. All this you don't find in meteorites. Okay, what do you find in meteorites? Maybe amino acids, maybe some fatty acids, maybe some organic molecules. You never see a protein. You never see DNA. Okay, all this is built by the system itself. There's a whole machinery. If you go a bit deeper now into this, okay, what is going on here, you see is incredible. Okay, so you have, okay, this is a metabolic chart in which all pathways, known pathways, are represented. Okay, so this is the general chart. If you go to E. coli, for example, which is a microbe, only a subpart of this, you see, I don't know if you notice the difference, only a subpart of this is carried out, but it's still a very complicated part in which cycles and linear pathways are combined, okay? And depending on the environment that you have, as Anais was explaining in the previous talk, different parts of this chart can be activated or deactivated, and microbes, they have a lot of horizontal gene transfer also to have, you know, if you go to an environment, they share what they know, let's say, from their evolutionary pathways to actually adapt to different environments and carry out different subsets of this general metabolic chart to actually construct all that complexity, and okay, this you heard from. I'm gonna jump, but let's say the domains in biology these days are three. We have a tendency to give a lot of importance to, of course, because we are animals, no, to animals, plants, but the fundamental differences at the basic level of microbes is between bacteria, archaea, and unicellular eukaryotes. Okay. If you go to humans, of course, humans are also carrying out a subpart of this chart. Okay. And not only that, but you know. Each cell in our human body, you know, is doing that, and at the same time, we have millions and millions of cells working in parallel. Okay, cell types, 200 different cell types in our body, doing, you know, being part of different tissues and different, you know, organs, and on top of that, we have a lot of bacteria working for us in the gut and all, and even more bacteria than eukaryote cells, the eukaryote cells that we inherit from our mother and our father, but just 2 kilos because they are much smaller. But the diversity and the number of microbes that we have in our bodies is so. Try to integrate all this. You know, it's just you know, it's crazy. We cannot understand this. It's you know, it's beyond our understanding. So, okay, I was gonna put a video here about the inner life of the cell that I recommend, but we don't have very good connection. So I just invite you to just take the inner life of the cell. I don't know how many of you have seen this film. It's a reconstruction. Okay. So some of you have already seen it. It's a reconstruction of what happens within any of our cells. Is impressive, and then you realize how complex we are. Okay, so the idea here is, okay, I am a physicist in origin. Okay, but I was interested in biology, and when you see this, you say, "Oh fuck!" I mean, I cannot understand anything. So, what shall I do? Okay, let's focus on the origins to see if I can see something there and try to understand. So I focus on origins because I want to understand life. Okay, but life, if you face it directly, is just, you know, So what we have to do is, of course, take into account biological knowledge, the fundamental facts of biology, okay, and try to see how we can implement them from chemistry, complex chemistry, okay. So the idea here is which are the two, I'm going to highlight two fundamental facts. Being a philosopher of science as well, fact, you know, that's why I put the inverted commas there. Fundamental fact, what are fundamental facts? But anyway, let's kind of, what are the things that we really have to take into account to approach the origins problem in a biologically relevant way, okay? One of them is the one that I already said, which is this. The synthesis of the key compounds is carried out by cells. Okay, you have this metabolism, the capacity of an autonomous production of those components that are going to rule the behavior of the system. Okay, so the actual rulers, the actual, you know, are compounds, are molecules. So we have to, but they are molecules that need, in order to construct those molecules, they don't come out spontaneously. You need a metabolism. So far from equilibrium systems, we are in continuous exchange of matter and energy with the environment. Why? Because there's an effort involved. There's a thermodynamic effort involved. It's not something that happens spontaneously, you know, on the planet. But life does it. Okay, we have to understand how life manages energy and matter resources to actually construct that incredible organization, okay, and those incredible compounds as well, proteins, DNA, RNA, all these big biomolecules that are responsible for their behavior, right? The other fundamental fact, let me go back to that. The other fundamental fact is, maybe you heard about evolution yesterday, is that all this complexity is the result of an evolutionary pathway coming from an original population of living systems that we call LUCA, last universal common ancestor. But it's not a cell. Okay, it's a population of cells. Okay, but through phylogenetic trees like the ones that Anais was showing before, we kind of project backwards. Okay, and try to see what kind of system did we have. Okay, but this is a fundamental fact, and it has been confirmed by, I mean, so many different ways of approaching the problem and methodologies, that the original insight from Darwin, okay, that all comes from a single origin, okay, has been incredibly confirmed. Okay, so there are no doubts that it's difficult to use the metaphor of the tree, and we have seen that maybe instead of the original tree of Darwin, we need to think about it in a network kind of way, or different ways of portraying the tree of life, which is no longer a tree in origin, but is maybe a bush or something. So we need some other metaphors here. But because it's very complicated, particularly due to this horizontal gene transfer problem, if you have horizontal gene transfer, phylogenetic trees become very, very complex. Okay, but if you also look at the biochemistry, we are going to go a bit deeper now into the biochemical features, a bit deeper than general biochemical features that we see in all living beings. We see, for example, that life doesn't use, for example, an amino acids. You use L type of amino acids. You've heard about this, homochirality, sugars, polysaccharides. D forms are used. So this is a kind of a signal that, you know, life selects part of the chemistry that is out there and takes it and plays with some chemistry, but it's kind of a selection of the chemistry, and a signature of that is homochirality. Also the fact that the genetic information is through DNA. We all have DNA, okay, and all that DNA is expressed into proteins through a common genetic code. all these points to, a single origin. So it's this molecular biology and biochemistry of the 20th century gave incredible support to the original idea of Darwin that everything came from a single origin. This combined with Pasteur, of course, and ruling out of spontaneous generation theories, plus other types of scientific knowledge coming from a proper calculation of the age of the Earth, because Darwin needed a very, very long time span, okay, and in that he died without knowing that the Earth was old enough, so to speak, okay, for his theory to fit in well. Okay, so all these pieces of scientific information have given an incredible support to the Darwinian picture. Okay, also if you focus on the energetics that are necessary to actually run all this, you see that all living beings use two types of chemical currencies, two types of energy currencies. One is soluble, chemical, and another one is chemiosmotic. Okay, so life uses gradients of protons and sodium ions coupled with chemistry, and also sources of ATP, GTP, this kind of. So it's all this is common to all life. Okay, you also have at the level of the metabolism that I was commenting before, you have a lot of intermediaries that are similar, and of course you have all life is cellular. All these features that are fundamental for life, okay, tell you that there was a single origin. Okay, and we can speculate about other forms of carrying out this kind of complex chemistry in other planets, whatever, but from the knowledge that we have about life on Earth gives us confidence that we are, origins of life took place is 3.5 thousand million years ago. Okay. Okay, so this was covered. So from these two fundamental facts of biology, when I finished my PhD, I proposed. So I'm from the Basque Country, and we are very bold there. Okay, and so even if I don't understand a thing, when I faced biology, the first thing I did in my research pathway was to propose a definition. Okay, I don't understand it, but let's start with the definition. So that's what I suggested. One fundamental concept was autonomy. This is related to this capacity for self-construction and these metabolic properties. And the other one is open-ended evolution, which is the potential for indefinite growth of complexity. Even if this could not be realized or not, we will see. There's a lot of debate on what is open-ended evolution. But anyway, so the idea here, the very important idea, is that we don't have to forget about. So under the influence of molecular biology, lots of emphasis has been given to the complex molecules that integrate biological systems like DNA, RNA, proteins. And of course, they are super important, and we have to understand more, and we have to be reductionist to try to understand more about them. But at the same time, there is a complex organization and dynamics of those molecules. So it's a game, it's a dynamic game that those molecules are involved in, with the metabolism supporting all that. Okay, so life is not something that you can ascribe to a molecule or a type of molecule. Okay, life is a game of diverse types of molecules. Okay, and so life is a systems property. Okay. And therefore, the origins should also be approached from a systems perspective. Maybe the problem of the field of origins of life is, again, under the influence of molecular biology, has focused on shortcuts to biomolecules, right? And if you were an expert in nucleotides, you would go for. If you were an expert in proteins, you would go, and the proteins came first, nucleotides came first, or lipids came first, compartments. There were schools that were fighting for their molecule, and in fact, what we need to do maybe is a different type of approach, in which we try to think about chemical precursors of those molecules, right? And see how they interact together previously, okay, to actually become increasingly complex, but taking into account some degree of organizational complexity from the very beginning. That's going to be one of my take-home messages here, right? In fact, this is a take-home message from a review that we published some years ago, in which we were saying, at last, we have the right platform in the 21st century coming from those chemists that are exploring complex mixtures of molecules and trying to see the emergent behaviors that are in those complex mixtures. At last, we have a different platform, not so reductionist, but more kind of trying to address the complexity of life from the complexity of chemistry. So what I say is from the complexity of chemistry to the hyper complexity of life, okay? Because life is hyper complex, right? So instead of searching for chemical shortcuts to the macromolecules, in particular the RNA molecules, where the RNA world, I suppose that you have heard about it, some of you, was the main hypothesis in the field since the discovery of ribozymes in the 80s. Okay, and so people were trying to go from chemistry directly, assuming that natural selection could be applied to molecules or populations of molecules, and not to proto-organisms. Okay, towards an RNA world from which DNA, RNA, and protein world could develop. Okay, this is being challenged these days. Okay, that's gonna. In particular, because RNA by itself if you just study RNA, you have a lot of problems, okay? They are crazy to find self-replicating RNA, and they are not finding it. And nature, you don't have self-replicating RNA, okay? So it's a kind of an obsession of us. We want to see molecules replicating, and we want to put them in the lab. That's not the way it works, okay? Also, there's the problem of the synthesis and the liability of RNA molecules. But so, particularly if you take naked RNA molecules, where do they come from? Okay, where do RNA molecules per natural selection go? Typically, they just fall into dead ends. In vitro evolution, we haven't all these people doing RNA evolution in vitro. They see that the molecules change in time. There is evolution. There is artificial evolution of the molecules, but there is no emergence of anything complex beyond that, okay? Typically, there's the simplification of the RNA molecules because the ones dividing more fast are the ones that win that game, full stop. Okay, you get into bottlenecks. Joyce and other people also did replicating molecules that could be like that in what he called an open-ended way, but there is no increase in complexity. There is just the continuous, evolution of those populations of molecules. Okay, so there are many things to address if you are taking just an RNA world position, and even if there has been some advances, very important advances in the synthesis of those molecules, like what you get from John Sutherland's lab here in quite a few years ago, there are big problems. And instead of having a pure RNA world idea, what we have to think, and this the work of Robert here is also from the bottom up, from a chemistry point of view. I want to mention Robert's work, and also from a top-down approach, I want to mention Lauren Williams' work, in the sense that we have to think is about nucleotide chemistry and amino acid chemistry coming together, right? And the evidence is clear from a chemical point of view. There could be connections there, okay, as they have shown us. And also, if you try to understand something like the ribosome, okay, and how such an incredible machine developed, you see that even if the RNA part is the one that has the catalytic core of it, and some people have claimed that's a sign that the RNA was, first. When you actually, these people doing structural analysis of the development of the ribosome, they see that, small nucleotides have to come together with oligopeptides. Oligonucleotides and oligopeptides have to there has to be a co-evolution or co-chaperoning effect so that you can build something as huge as a ribosome. So there's evidence coming both top-down and bottom-up, saying that that is not simply RNA that we have to deal with. Okay, so instead, okay, my view. How am I doing with? Okay, still plenty of time. No? Okay. My view. This is still the first part of my talk. Okay, so my general view, because I'm just giving a general view, is a situation in which molecular and organizational complexity develop hand in hand. Okay, so you have to start with something very simple at the molecular level, but this kind of basic autonomous systems, minimal metabolism. I will speak a little bit about what is minimal metabolism in a minute. Then go to hereditary autonomous systems where you have one or two polymer world, two oligomer world, let's say, okay, with oligonucleotides and oligopeptides here developing, and then you can have some kind of heredity in the system, and then you would go to another stage in which you would have, minimal living systems where there's already kind of a genotype-phenotype decoupling and a code and translation apparatus and all the rest of it. But these are huge transitions. Okay, here I'm representing what happens at the level of the individual and at the level of the molecules involved. But it also, as I will say at the end of the talk, it also involves dynamics that happen at the ecological and evolutionary level, as I will. So instead of having this picture that I was having here, in which you were trying to shortcut to biopolymers, the idea here is that we have to compartmentalization and metabolism and the capacity of organizing a set of molecules in a unit, okay, in a protocell, much more importance, okay. And the community of origin of life researchers are realizing that this is the way to go, so to speak. So we start from something that could be self-organizing network in which you have feedbacks, positive feedbacks, negative feedbacks. I think Robert Pascal was speaking yesterday about autocatalysis and networks and in chemistry. Okay, the prototype was Belusov-Sabotinsky reaction, where you had complex behaviors and you could have macroscopic patterns, spatial and temporal patterns. The problem with the Belusov-Sabotinsky reaction is that it happens at pH almost 0, and the chemicals involved are not prebiotic. Whiteside's lab, they were doing similar things, like combining much more metabolically and prebiotically relevant compounds, in which you could see self-organizing dynamics. Okay, generation of patterns, oscillations, complex chemistry with components that are relevant prebiotically. Okay, so that was an important paper in which they showed that self-organization could be a first step, but as I was saying before, it's not simply self-organization. Self-organization is like a spontaneous pattern, generation of a pattern, okay, out of a combination of many molecules coming together with reactions. There has to be positive feedback, so a small perturbation, a small microscopic fluctuation becomes a long-range correlation in the Prigogine terms. Okay, so Prigogine showed how you could generate these patterns of behavior, complex patterns of behavior, starting from very relatively simple chemistry, but provided that there was an autocatalytic, like a kind of a positive feedback that could generate this long correlation, and then a negative feedback that could stabilize the pattern. Okay, so it's a combination of feedbacks that you have there, and you can have complex chemistry. But life uses self-organization, somehow it kind of is based on self-organization, but it goes way beyond self-organization because it has to generate. So self-organization would be like you have things, complex mixtures, they come together into patterns, okay, spontaneously, but the work that living systems do is much more than that. They have a metabolism to actually, produce stuff. There is not self-organization, it's self-construction, literally. Okay, self-production. Okay, these are topoietic terms. Okay, so the good news, and I will finish this part with a very quick review of some let's say papers that have been there for the last let's say 5, 7, 8, well, there's decade or something. The good news is that there are these chemists that are interested in actually addressing metabolism. Okay, and I can mention here the work of Sutherland again, because he did not only contribute to understanding how an RNA molecule could be synthesized under prebiotic conditions, but also he showed that hydrogen cyanide chemistry could lead you, from that type of chemistry, you could get the building blocks, okay? Very different building blocks, amino acids and glycerol, the nucleotides that he would. So a diversity of building blocks that were relevant for life came from the same type of chemistry. Okay, so there's a kind of a connection between a type of organic chemistry and the chemistry of life. Okay. He's been criticized for doing one step at a time, and there are lots of cross criticisms here that I will not go into. But that I think is important. For example, there's a lot of criticism. I was speaking with Robert this morning between Sutherland and Ralser. Ralser is another researcher who has also proved that metabolic pathways, current metabolic pathways, could run without enzymes, because one of the things that classical researchers in Origins of Life, like Leslie Orgel and all these guys, were saying is that, okay, very nice to have the metabolism first approach, but we need enzymes to actually run those pathways. And there is a bunch of labs, like Ralser's lab, that is showing that metabolism could run under alternative catalysts like iron or other type of inorganic catalysts, and you could run fundamental metabolic pathways without enzymes. And this is fundamental to open our minds and try to explore that. Okay. Also, Pauner in United Kingdom. Glycolysis without enzymes again, or Moran's lab in Strasbourg, where starting from pyruvate and glyoxylate, they get a suite of other compounds that are remarkably close to the TCA cycle, which is the Krebs cycle. Well, it depends, the reverse TCA cycle, depending on whether you are under oxygenic or non-oxygenic conditions. But the idea here is that you start with 2-3 compounds, and you generate a mixture of compounds that are the actual compounds that something as fundamental as the TCA cycle runs upon. So the idea here is to get closer and to try to find bridges between organic chemistry and biochemistry and the metabolic pathways. And I want to mention also here, even if there is also tensions between Ram and Joseph Moran, but Ram Krishnamurthy in the United States also is doing very interesting work with this glyoxylate paradigm that Eshem Mosher actually was the guy who gave the idea to Ram. And the interesting thing that Ram is doing is not trying, maybe Joseph is trying too much to kind of mimic exactly the pathway that we see in living organisms. Ram is taking a very intelligent approach, which is to get to the vicinity of the TCA cycle, not directly to the TCA cycle, and exploring the chemistry around the TCA cycle and hoping that the catalyst and the metabolic pathways will co-evolve to actually finally get to the TCA cycle. I think it's a very, very intelligent strategy, not to try to go directly and mimic what we see in life, but try to approach it at different stages and letting then evolution of the catalyst and the metabolic pathways lead you to the actual what we see in biology. Okay. Other proposals, maybe more theoretical, in which they try to think about a type of metabolism that was not so based on phosphate, for example. Okay, thioesters, so stuff like that. Okay, we should try to conceive of potential proto-metabolisms or minimal metabolisms that could run under, could be a bit different from biological metabolisms. And so there's a lot of exploration of these days, and seeing all this flourishing of approaches to try to address the origins of metabolism. We recently wrote a paper, kind of a review paper in bioessays, where we were kind of claiming that the idea of metabolism should play a role at the interface between complex chemistry and biology. Typically, when you think about metabolism or minimal metabolism, is the chemistry that supports life. Okay, but then the concept of metabolism depends on the concept of life. Okay, and what we need is to kind of try to find criteria by which you distinguish a metabolism from a set of reactions, a network of reactions. Okay, but also distinguish it if it has to play a role at the interface. Okay, and before we get living systems, you cannot make the concept depend on the definition of life, because life is here, right? So you have to think about how to characterize a minimal metabolism from chemistry towards biology, but going beyond chemistry and not using something like a definition of life. Okay, so the idea here also is that, maybe Robert said something about this yesterday, how in this process chemical diversity gets narrowed down so life doesn't use all the organic chemistry or all the chemistry that is available. You have to actually do a selection of the chemistry to play a game that is going to become hyper complex and is going to give you another window of dynamic behavior, but not with all the diversity that you have. You have to select in order to move forward in complexity, and what is called functional diversity is not chemical diversity anymore, it's functional diversity, as we will see in a moment. Okay, and this comes hand in hand with the capacity of these systems to gain autonomous control of their own dynamics. Okay, so initially maybe the environment was more influential, and later on these proto-organisms, these proto-cells, were gaining autonomous control progressively as they were becoming closer and closer to organisms proper, not proto-organisms, but organisms. Okay. In any case, it's good to finish this part. It's good to see what we can get at this level from top-down approaches. And I don't know how many have read about Craig Venter's mycoplasma. No, you don't know about this. Okay, Craig Venter, yes, no? Craig Venter, and he's a very famous scientist. He's maybe too famous, and he died in his own success because he did something in 2010, and he said, I'm the new, and then nobody paid attention to something much more interesting. Well, no, nobody now, but 2016, he really did a minimal. So in 2010, he copied Nature. To make a long story short, 2010 he copied Nature with incredible skills, methodological skills, by copying Nature and creating a hybrid. 2016 he really did a brand new, let's say, organism. It's half the genome of mycoplasma. So. And this is giving a very interesting platform at the level of a minimal organism to try to understand what's going on there, and seeing, for example, the metabolism that is this essential metabolism that typically people would think about minimal metabolism as this metabolism that these cells carry out. But of course, the level of complexity of that metabolism is you have RNA, DNA, proteins there, okay? Because you are starting from life, and you are just by gene knockdown strategies, you are getting, you know, all those genes that are fundamental, and not others, but some could be missing, for example, for a proper reproduction, and these people have said that this was not properly reproducing properly, and you need to add a few more to actually have reliable reproduction of these systems. But it's very important that we have I think that even if I don't like Craig Venter too much, it's very important that someone does this job, okay, from a top-down approach, and gives this platform also to understand minimal life at that level, okay. And there's recently, in 2022, there's it's also a Cell paper, very interesting, where they are simulating all that at that big scale, okay. So we have also capacities to simulate things that, you know, involve so many reactions, so many proteins, so many metabolites, and but I'm not, unless we want to go here in the discussion, I'm not going to go into these details, because otherwise I will I will not finish today, but tomorrow. Okay, so so the idea behind our approach of minimal metabolism is that you need to have a network of reactions, but if you stay here, this is not going to give you enough to actually characterize that as a metabolism. So typically people think about the metabolism when they are thinking about this network of reactions that supports the system, okay? And the constraints level here in a real biological system like this one, you see all these here in mycoplasma, right? So you have proteins, you have DNA, you have ribosomes, you have, so I mean, all these complex molecular mechanisms are synthesized, and they are constraints on the actual metabolism that is running. They control spatially and kinetically what's going on, so in terms of space and time, you know, and also energetically, of course. So there are, the idea here is to abstract from that and say, okay, we need the synthesis, but also the control part. So there's a functional bootstrapping here between a reaction network and those products of the reaction network that take control. And here you can think about autonomous control, because you are actually synthesizing the stuff that controls the reaction. So there's a feedback between, you know. So that's what I try to tell now people working on origins of metabolism, that they should not just simply go and see what are the reactions that give you the intermediaries or whatever like that. You have to run this under non-equilibrium condition, which is a technically difficult part of it. But also you have to have in the same pot, so to speak, not only the reactants, the metabolites, but also the products of those reactions that actually have a catalytic effect or a spatial effect on the actual network. Okay, so you have to start organizing, and when you have some signs of this spatial and temporal organization that is self-generated by the system, there is where you have to say, I have a minimal metabolism, because just a network of reactions won't make a minimal metabolism. Okay, I'm going to run fast, but because it's already 12. I'm watching. So the idea here is that we have we were using a truncated cone as a graphical metaphor to actually say that it cannot be a cone just going to one type. So the idea here is that there has to be diversity at the level of the molecules carrying out the reactions, and there has to be diversity at the level of the constraints as well. So you have to combine different types of supramolecular structures, oligomers, maybe some kind of prebiotic energy currency to actually drive the whole thing, because we need to deal with, you know, spatial control, kinetic control, energetic control, and variability control. But so we use here, instead of a truncated cone, we use the tetrahedron metaphor to actually say that maybe origins of life, you can shrink the problem and not deal with all the complexity of life, but the shrinking of that problem cannot go all the way to a point, so to speak. You know, a non-reductionist approach to origins of life, you end up with a tetrahedron, but you cannot go further. If you simplify further, the idea here is that if you simplify further, you are missing the window of opportunity to actually go from chemical complexity towards increasing complexity, the window of dynamic behavior that would open if you combine different types of constraints. Okay, you understand the idea? By the way, I'm speaking about this. Okay, I should have said at the beginning. If you want to interrupt me, you can. I actually I'm getting tired now after 1 hour. So it's like when footballers play more than you have to do extra time. Then the typical lecture time is 50 minutes. Now I'm getting tired. Like the muscles are here. My muscles here. I'm gonna make a kind of a break because I'm gonna introduce some videos now in a minute. But I've been claiming that this combination of constraints is very important, and in particular, the fact that you have a container. Okay, that's why I'm a specialist in protocell models, in vitro and in silico. Okay, but one thing is that you say it yourself, and it's much better than a Nobel Prize like Jack Szostak says it. So Jack Szostak said, although protocellular structure poses more problems initially, it is actually simpler to solve these problems upfront, rather than leave them till later when they could become completely intractable. So that's what I was saying after my PhD student for my PhD dissertation for many years. But it's much better that someone like Szostak says it, because then people pay attention, So I'm going to skip a lot of slides now because I have maybe as many or more than the ones that I've shown so far. So I'm sorry about that. The videos are going to be about this. Now I'm going to focus, and I'm going to do it very, very fast because I don't have time. I'm going to focus on what type of systems approach I have developed scientifically, okay, from this philosophical or conceptual view, right? And you can see the papers. You can, if you're interested in this, you read, and then I'll be available here or when I go back to the Basque Country. But the basic idea is that here, what are you seeing? Just fatty acid in water, okay? And they create these beautiful structures, okay? They are supramolecular structures that could be the scaffolding around which, if you couple this with chemistry, you could have interesting protocellular behavior happening, right? But these are fatty acid vesicles. You cannot think, if you think about, now I'm going to give you the modeling part of my work is based on using not the phospholipids that make the membranes, biomembranes, right? But a chemical precursor, as I said for the other things, we have to think about chemical precursors of biomolecules. And also the phospholipids are too complex, okay? So I'm going to be using fatty acid like in this cryo-electron microscopy image, right, that we got in our lab. And but this is the I think fatty acid you find in meteorites, you find it out there. So let's start with this is one of, and fatty acids are the basic from which you develop some types of phospholipids. Not all phospholipids, as you will see in a minute, but so a biomembrane is absolutely nuts again in terms of the complexity it conveys, right? You see all these colors here? It's a joke, okay? Again, it's a joke, just a few. Because in reality, so you have membrane proteins, yeah, and of very diverse types. But in terms, if you just focus on the lipids, the families of lipids, I mean, you cannot really, I mean, it's incredible. You synthesize phospholipids, each of your cells, each of your cells is synthesizing different types of, let's say from a mammalian, now it's genomics, proteomics. Have you heard about lipidomics? Okay, so if you actually see the families and families and families of lipids that you have in one single membrane, right? If you take away the membrane proteins, just the phospholipids, it's just crazy, okay? So from modern cellular boundaries, we are thinking of getting a protocell starting from vesicles from simple amphiphiles. So simple amphiphiles, where do I have my, yeah, right. So you see here, this is a phospholipid. This is a precursor fatty acid, okay? And we are working, me and Szostak and many people are working with fatty acids because they are relatively manageable under lab conditions, right? So the idea here is there are many differences in the properties of the membranes when you have phospholipids and fatty acids. I'm not going to go into all these details because I'm going to really, I don't have time in any case. But I'm going to show you some videos. Okay, let's see if this run. Let's see if you see it. Okay, so here, for example, one of the big differences is that these are fatty acids, okay? And then the other little things that you see are other compounds. But then one of the things that is different from a phospholipid membrane and a fatty acid membrane is that there is a lot of, in fatty acids are more soluble in water, okay? So there is more monomer in solution, and the dynamic exchange between the supramolecular structure and the monomer in solution is much more, is present, and you have to take into account. If you have phospholipid, basically there's no phospholipid in water, therefore you have the supramolecular structure and stacks there, right? This is very interesting for origins of life because having a more dynamic boundary initially is much more interesting. When you go to a phospholipid membrane, you need membrane proteins to actually have these dynamics going on. And the bilayer is a matrix that is more impermeable, but initially it's much more interesting that it's dynamic and it's it's more permeable because you don't have advanced mechanisms to actually deal with matter and energy. So you see here how the monomer gets into the bilayer and then flip-flop. When you see the ones in white, it's just the same fatty acid, but doing flip-flop, okay? So this is one of the videos. This you can download. You can see in SOSTAC lab. So then there are more there, but I'm going to show you just a few. Okay, see, you can make these vesicles grow. It's very difficult to make a phospholipid liposome made of phospholipids. It's very difficult to make them grow. But here, if you add micelles or you add fresh lipid to the medium, you can make these supramolecular structures grow more easily. So there's advantages of working with chemical precursors, and this is something that we also found in the work that maybe I mentioned if I have time, that I did with Robert, in which we were using phospholipids to actually. Robert gave me the chance to collaborate with him some years ago, and he had a reaction that was taking place in solution, and we put vesicles of fatty acids and vesicles of phospholipids, and the vesicles of fatty acids gave some interesting effects that the phospholipid vesicles didn't give. And so sometimes working with precursors is an advantage to the chemistry that you are exploring. Okay, so that's one of the things that you can do here is to try to make these systems grow and potentially divide because they are much more dynamic. Okay, this can be done with micelles. You see the micelles there approaching there. They're different, and then these are the vesicles. Okay, and this is a way that SOSTAC was using to actually make them grow and divide. Okay. And the other video that I wanted to show you is the one in which you can also prepare mixtures. Here you don't see mixtures, but SOSTAC was using, in the video you don't see the mixtures so much, but here you have a different type of, now it's a nucleoside that is going to be, so SOSTAC wanted to have nucleotide replication inside the vesicle, okay? And he prepared vesicles of fatty acid, mixed vesicles of fatty acid to modulate the permeability so that the building blocks for the RNA or DNA the oligonucleotide that was going to form inside the vesicle, the monomers could go through, but the actual polymer or oligomer could not get out. So you can modulate also the permeability properties by using diverse, like preparing the vesicles with the right composition. Okay, so we start from here. You see there's some simple chemistry going on, simple chemistry and self-assembly processes coupled, okay, they happen inside much better. So the idea here is that functional relationships are going to be developing in this context, okay? So you're going to have a number of constraining components, including the boundary itself, that is going to be That's going to be, you can tell that they are functional because they contribute to the maintenance of the system, of those systems. Okay, so systems, you're taking the system by coupling these supramolecular structures, the self-assembly that are quasi-equilibrium structures. With chemistry, you can take the system away from equilibrium, okay, and you can make it grow and divide potentially, okay. So that's the idea here is that we start with function as a fundamental concept, and we actually have philosophy behind it, like reflecting on what is function from a point of view of. So that's what philosophy comes from. Philosophy also has an input here, because we reflect a lot about what is function in biology. And one of the important things is that when you're thinking, imagine that you're thinking, oh, I have a catalyst. I found the catalyst of a reaction. Oh, it's super good. It has a function. Don't pay attention to those people that tell you that single molecules have a function. Because what they are doing is a trick. They're saying, okay, the function that a catalyst like that has in a cell, by contributing to the maintenance of the cell, in the cell, now you are making the trick of saying, I have a function here. The function will only be there if that component is part of an organization, and that component is contributing, as I would say here, specific contribution of the distinguishable part of a system to the maintenance of the system. That's a physiological way of understanding function in biology. There's another one that is related to evolution, but I will not go into the details of the discussion here. But that, as an example of how conceptual reflection also helps in the way you put science in context, right? But the important thing is what I said before. You have to have this organizational, I don't know if you can read, organizational dimension and evolutionary dimension. Okay, and the idea here, if I use the pointer as I should, here is that you're going to have different evolutionary dynamics depending on the individuals carrying out the dynamics, right? And if you change the type of individual, the evolutionary cone is going to change until you get to individuals that are reliably reproducing and all that. Okay, so we have to envision the whole process of origins of life as some transition stages where protocells resembling more and more cells developed, but in the context also of phylogenies that were developing relation, you know, so it's where reproducing systems, maybe not so reliably initially, but then as hereditary mechanisms were developed, it would do, and that's what we should do. And the idea here is that initially the environment and maybe the mixing and the fission and fusion events here were driven more through environmental forces. As you start developing minimal metabolism and you become as an individual more autonomous, then you have families, more defined families, because reproduction is more reliable. You can have families, not just the mess that you have initially here. There is a funnel here that represents that chemical diversity. Somehow you see here molecular organization complexity, diversity here, chemical diversity of protocellular chemistries. You see the funnel represents that the chemistry is selected. So natural selection should be playing a role here in these families of protocells, right? And less and less, the influence, there are environmental influences here, of course, but natural selection takes a increasingly more prominent role as these individuals become more and more, you know, elaborate organizations until they reach something like LUCA. And here, but here there is ecology. There is in these families of protocells, there should be ecological relationships as well, and evolution is playing a role here. And you from a stage in which fusion was more important than fission you had here, but then reproducible fission was more important as you go here, and you have here exogenous driving forces and endogenous functional diversity. So it's kind of, there is a transition between systems that were initially controlled by environmental conditions to systems that were increasingly autonomous, right? Until you get to that. With that, and with an acknowledgement to the PhD students that here there's a room full of PhD students. I want to say thank you for giving me motivation, right? It's working with people that gives me motivation. And my PhD students here, Pietrafita, Certedis, Sara, and Nino now, are the pillars of the work that I didn't show, but next time. Okay, thank you.